Hi! Today we're talking about nucleic acids. Nucleic acids are going to be one of our other organic macromolecules. And so by organic, what we mean is that it does contain at least C, H, and O. That's what it means to be organic. They are going to also have nitrogen and phosphorus. So those are the atoms, those are the elements involved in building a nucleic acid. These 5 elements are going to be arranged into a subunit called a nucleotide. And when we looked at carbs, the subunit was a monosaccharide. When we looked at lipids, we had the glycerol and the 3 fatty acids. When we look at nucleic acids, the subunit nucleotide actually has 3 distinct parts. And when we start putting these together to form a polymer using DNA polymerase, the enzyme, or RNA polymerase, the enzyme, we see that these 3 parts really come into play because they're going to form a sugar phosphate backbone, sugar phosphate, sugar phosphate, sugar phosphate, with these nitrogenous bases hanging off. And so we do want to focus on the sugar and the phosphate. Important thing, this is the phosphate group. This is the functional group. This is negatively charged. That'll be really, really important when we get to biotechnology and manipulating DNA. Then we have a 5-carbon sugar. And then we have a nitrogenous base. So the sugar is going to be the CHO. Here we've got the phosphate and then the nitrogen here in this ring. One important thing about this sugar is actually that the sugar comes in 2 forms. So our nucleotides are basically broken up or classified in 2 different ways. Some nucleotides differ in their nitrogen base. Some nucleotides differ in their sugar. The sugar, and this is where the names DNA and RNA come from, deoxyribose, and then just ribose. So the one on the left is deoxyribose, the one on the right is ribose. The main thing is that these are going to be functionally different because the shape is different, because we are missing an oxygen actually right down here on the bottom right. You don't need to know where the oxygen is missing. You just need to know that they're structurally different, and therefore they are functionally different. They are going to end up performing very different functions. And all of it has to do with the shape and the bonding pattern of that sugar. One thing that we do want to note, and it's kind of hard to see in this picture, is that in this sugar, the carbons are going to be numbered. So it's a pentose, and the 5th one actually hangs out here at the top, and the 3rd one is down here. We'll be talking about 5' and 3' carbons in there. So we've got the negatively charged phosphate group. We've got the 5-carbon sugar that comes in 2 forms. Then we've got the nitrogenous base. The nitrogenous base comes in 5 forms. 2 of them are called purines, 3 of them are called pyrimidines. So let's look first at these purines. This is your adenine and your guanine. So you probably remember this from your introductory bio class, A's, T's, G's, and C's. But now we need to actually know the molecular structure. The important difference between the purines and the pyrimidines is that purines are double ring nitrogen rings, and pyrimidines are single ring. Structural differences create functional differences, and we're always trying to keep that in mind. One shortcut that I've heard and that I use, if you remember chemistry, Ag is the abbreviation for silver, so you can think pure silver, A and G, adenine and guanine, pure silver, Ag. And then you just need to remember the rest would be pyrimidines. Now the rest is actually CT and uracil. The reason that we have to have uracil is that thymine cannot bind to ribose sugar. And so uracil ends up taking its place. You should actually be able to kind of identify the difference here. We are missing a what's called methyl group or single carbon out here. That's the difference between thymine and uracil. You don't need to know the difference, but you should be able to identify the difference. So we've got 5 possible nitrogen bases. Thymine is only in DNA, and uracil is only in RNA. So when I'm looking at DNA and RNA, they only at one time ever have 4 possible nucleotides. Deoxyribose is found with AGCT. If I'm looking at a ribonucleic acid or a nucleotide in ribonucleic acid, the only possible nitrogen bases are A, G, C, and U. So we do want to know what possible bases are found in bonded to a deoxy or a ribone. And then we need to know the basic structure. The other thing, and we'll talk more about this later in the year, but we want to introduce it now, is that these will actually do what we call complementary base pairing. Now, most people would think that base pairs are only in DNA because we know that DNA is a double strand. The DNA does not stand for double, but it's an easy way to remember it. DNA is always a double strand of deoxyribonucleotides. And so we want to kind of look at this complementary base pairing here in DNA because it's easy to see. But complementary base pairs are found in both DNA and RNA. And these base pairs are what allow DNA and RNA to do their function, which we'll talk about in just a second. So DNA stands for deoxyribonucleic acid. You've seen pictures of DNA forever, but this is the one that you're going to need to be able to analyze and interpret in AP biology. A lot more detail here. You should be able to find the sugars and the phosphates. We do want to go ahead and notice that here the oxygen is pointing up, the phosphates are pointing up. Here they're pointing down. This is a concept that we call anti-parallel. The sugar-phosphate backbones are running in opposite directions. So of course you know DNA is double-stranded. But now we want to know that when we put each nucleotide, right, you should be able to find one nucleotide, 3 parts, phosphate, sugar, any nitrogen base. When we polymerize and put these monomers to make a polymer, the backbones actually run in opposite directions. The other thing we want to start noticing is the single rings, the pyrimidines, T and C, and the double rings, the purines. But the other thing we notice is that there is always a pyrimidine bonded to a purine. And this actually gives us a uniform diameter. There's always a larger nitrogen base bonded to the smaller nitrogen base. And here are these hydrogen bonds. We talked about hydrogen bonds with water. What causes hydrogen bonds? Well, polarity. These nitrogen bases must be slightly polar, and it's that polarity of the nitrogen bases that is actually making these bond to each other. So we'll get much more into this later. We'll talk about how many hydrogen bonds. It's actually A and T form 2. We get out of school at 2. G and C are going to triple bond, triple hydrogen bond, and that's going to be year 3. Keeping in mind that hydrogen bonds singularly are very weak, but when we have a lot of them together, they're very, very strong. RNA, you've got your 3 basic RNAs that you've talked about for a long time. This is ribosomal RNA, or rRNA. B is transfer RNA, or tRNA, and it transfers an amino acid. which we'll talk about more when we get to proteins. You should kind of remember this shape. And then your last one is messenger RNA, or mRNA. And it's really easy to see the nucleotides here in C and B, but A is actually nucleotides, ribonucleic nucleotides, plus protein. And this brings about the chicken and the egg a little later. How do you make protein if you don't have RNA? How do you make RNA to make protein? That type of idea. We'll talk much more about their functions later. There are other forms called small interfering RNA, siRNA. There are what are called SNRPs. There are all sorts of different RNAs that are still actually being discovered. So you should definitely start to learn these three types, and we'll talk about more later. So we want to know that DNA always has one common structure and start learning things about the structure. And then we want to know the three different types of RNA and that they have different jobs. So the last part of the notes is about what are their jobs? What do they do in a cell? Why do all cells have DNA and RNA? And this is a big idea called the central dogma. And central dogma basically just means main idea. And the main idea is here. Now we have the DNA, we go to RNA, and we go to protein. So DNA actually has the code to build proteins. And we'll talk more about this later. So here it says information. We want to kind of fill in code to build proteins. Now part of the central dogma, the main idea, is also that we can copy DNA. We can replicate it so that every cell, and as cells divide, they all have DNA. But we really focus on this transition of the information to build proteins is held in DNA, but we use a process called transcription that we'll get to later to make or synthesize RNA. And then RNA is actually what's going to go to the ribosome. So if you remember from Bio 1, we've got all of these steps to making a protein. And we're going to focus on proteins later. The DNA and RNA kind of carry the information and express the right parts of the information. That's the big thing we'll talk about in this class. It's not just this process, but how to control it. Which cells are making keratin for hair and nails? Which cells are making melanin? Which cells are making insulin? And so we'll talk about that later, the proteins and how to control this. But we really want to know that DNA kind of is the keeper of the information, and RNA is actually doing the work to get this protein made. And so what we have to be able to do is send information, and this information is all in what we call the sequence of the nitrogen bases. So if you remember a sequence AATA is going to give us a different protein than ACGG, and etc. And so it's the sequence of these letters that will help us build a protein, and different proteins do different jobs. So that's where we are for now. If we look at the review questions about nucleic acids, we need to know what is structurally important about a nucleotide. What is the structure? How are they different structurally? How are they alike and similar? What types of bonds, these hydrogen bonds? And then how are the jobs of DNA and RNA different? So the keeper versus the transfer.